In this figure, we're going to get into the topic of nitrogen metabolism. Coming up is a very complicated figure dealing with nitrogen metabolism, but before we get to that, what I'd like to do is to say a few introductory comments, which brings up a very important concept. No matter what amino acid that will be discussed, no matter what the amino acid is, the first thing your body does in catabolizing that amino acid is to remove the amino group. By removing the amino group, we end up with two products. One product is the carbon skeleton, and the other is ammonia. Now, if you hear me say the word ammonia or amino group or nitrogen, I'm referring to this NH3, which, as you know, is highly toxic, especially to the brain, causing cerebral edema. Now, the carbon skeleton, what's the body going to do with that? The carbon skeleton can take that carbon and burn it as a fuel in order to get ATP. Alternatively, the carbon skeleton can be used as a precursor to make other kind of molecules, such as epinephrine. The amino group, on the other hand, the body wants to get rid of it. Why? It has no energy. So by removing that amino group, it wants to dispose of it, get it out of the body. And how is that ammonia going to get out of the body? It can get out of the body in two forms. One form is urea, made by the liver. Other form of getting rid of that nitrogen is in the form of ammonia or ammonium ion itself, and that is disposed of by the kidney. So that is what will be discussed first. How does your body deal with that nitrogen? That's why this topic here first is nitrogen metabolism. After we talk about the nitrogen, then we'll get into the issue, how does your body deal with the carbon skeleton? Now, regarding the enzymes which carry out the first step of this catabolism, the removal of the amino groups, that class of enzymes that remove amino groups from amino acids, they're classified as transaminases. Now, all transaminases, and if you have 20 amino acids, that means at least 20 different kinds of transaminases out there. Don't go worrying about individual names, just know the surname transaminase. All transaminases require a particular coenzyme, and that coenzyme is pyridoxal-phosphate, which comes from the vitamin B6, also called pyridoxine. So pyridoxine is a vitamin used to make the coenzyme called pyridoxal-phosphate. Notice that suffix in pyridoxal-phosphate, that Al, is telling you there's an aldehyde group, is the business part of the molecule. Aldehyde groups bind to amino groups, and in this case here, the amino groups of the amino acids, to take the amino group off of an amino acid. Now, another issue to talk about here is, what is urea in the first place? No one expects you to know the exact structure of urea, but in my opinion, it's important to know what it is. It helps you understand what the body's trying to do with that nitrogen. Now, what urea is structurally is simply two amino groups linked to a carbonyl. This is urea, and it's going to be made by the liver. Now, a very, very important issue comes into play here. The liver is going to make this molecule called urea, the structure you see here. And after it makes it in the urea cycle, it's going to dump urea into the bloodstream. When urea gets dumped into the blood, what happens to the pH? Does the pH increase, decrease, or no change? And the answer is that pH does not change. And why doesn't it change? Because these are amide nitrogens in urea. Your body knows chemistry. It knows that amide nitrogens are never, ever charged. So what it's going to do is take this amino group, which if the amino groups get dumped into the blood by themselves, then, because ammonia is alkaline, as a base, and ammonia will pick up a proton from water to form ammonium ion, and OH negative results. OH negative is alkali, raises the pH. Your body knows that chemistry. It knows ammonia is a base which would raise the pH. So what your body's going to do is take that ammonia and build it into this thing called urea, the structure you see in front of you there, and urea is dumped to the blood and does not change the pH. It's a beautiful compound in the sense that once it's made, it will not change the pH. Not only that, urea is sufficiently nonpolar. It goes right through membranes. There's no transporter for urea across membranes. And not only that, it's sufficiently polar, ironically, sufficiently polar when it's dumped into the blood. There's no carrier for it, like albumin. It doesn't need a carrier. It's sufficiently nonpolar, sufficiently polar, and non-toxic. It is an amazing compound, that thing here called urea. Now, one last item here before we get into the metabolic pathways here regarding nitrogen metabolism, and that is this word urea. Don't confuse the word urea with another word which sounds very similar to it, and that's the word urate. These are two different kinds of molecules. Urea is not urate, even though they look very similar. In fact, both of these molecules there, urea and urate, they're not the same. Both of them are end products of metabolism. Urea is the end product of nitrogen metabolism. Urate, also called uric acid, is the end product of purine metabolism. But they have nothing to do with one another. The names sound the same, but they have nothing to do with one another. Okay, now with that introductory comments out of the way, now we're going to get into this very complicated figure that you see in front of you. Now, the way I'm going to approach this figure is I'm going to start at the bottom of the figure first and talk about this ammonia and urea. Where does that ammonia and urea come from in the urine? And start with that point there. In the urine, where does that ammonia and urea come from in the next figure? So this is the bottom of the figure that you see in front of you. So here at the bottom here, this is the urine, and in the urine you have urea and you have ammonia. Now, where do they come from? They came from the kidney. Now, regarding the urea is the major excretory form of nitrogen. When you get rid of that nitrogen, most of that nitrogen is being excreted in the form of urea. 86% of all that nitrogen that you excrete out every day is in the form of urea. Now, what about the ammonium ion? That constitutes just very little percentage-wise. Only about 3% of the nitrogen that you excrete out of your body every day is in the form of ammonium ion. Despite the fact you spend a lot of time in renal physiology talking about the ammonium ion, and in the kidney and how it works, this is the amount that you excrete is very little. Now, I know you add up those percentages here and you say, hey, that's not 100%. That's true, and the reason is because you excrete other kinds of nitrogen in the form of creatinine and other kind of molecules which constitute the remaining percentage to get that 100% that's excreted per day. Now, we're going to start here with ammonia. Where did that ammonia come from that the kidney peed out into the urine? Where the ammonia comes from is in most tissues. In most tissues, there are a variety of deamination reactions that go on. Not just of amino acids, you have other kind of deamination reactions going on. For example, there's an enzyme called adenosine deaminase that's discussed in chapter 18. That enzyme pops the amino group off adenosine, and there it is right there. But the ammonia can come from other sources as well inside the cell. Now, that ammonia, once it's popped off carbon, that ammonia, again, your body knows, don't dump that ammonia into the blood because the pH would rise. So your body's smart. It has a very low yield enzyme. Don't go worrying about the name of the enzyme. But the enzyme, what it's going to do is take the ammonia and put it onto the R group of glutamate. Glutamate has a carboxyl group on the R group, puts the ammonia on the carboxyl group and making the amino acid glutamine. Now, why is that significant? Because if you take ammonia and put it on a carboxyl group, that's an amide bond. Remember, amide bonds are never, ever charged. So what your body's doing is it knows that chemistry. So it knows to make this glutamine. Glutamine gets dumped into the bloodstream. Once it's dumped in the blood, it will not change the pH because it has an amide nitrogen. And it's bringing to the kidney that nitrogen that the body wants to dispose of. And in the kidney, the reverse reaction takes place. There's another low-yield enzyme. Don't go worrying about it for the boards. But this enzyme is called glutaminase. What the enzyme is going to do is just do the reverse reaction, just pops the amino group back off, regenerating the glutamate. Now, the ammonia will pick up a proton to form ammonium ion, and then that is then eliminated in the urine. That's where the ammonia comes from in the urine by that deamination reactions and moving it through the bloodstream in the form of glutamine. So now, what's going to happen next? What can happen next? So here's the glutamine getting into the blood, can go to the kidney. Now, that glutamine, when it gets into the blood, right here, can also go to another organ, which is shown in the next figure here. Here's that glutamine that's coming from most tissues. But instead of going to the kidney, it can also go to the intestine. When it gets into the intestine, that same enzyme that was discussed in the kidney is also present in the intestine, called glutaminase. Now, what's going to happen to the glutaminase? What it's going to do is remove the amino group from the R group of glutamine, regenerating glutamate. But then here's that ammonia. And then ammonia right there in the intestine can also come from intestinal bacteria. The kidney and the intestine just absorbs it. But regardless, here's the ammonia that the intestine has. It dumps into the blood. Now you might say, isn't that going to be a problem? It's not going to be a problem. Why? Because right there is the liver. The liver immediately removes that ammonia. Otherwise, if it wasn't for the liver, that would be a big time problem. But simply the ammonia is removed quickly by the liver out of portal blood. And that ammonia right here, when it gets into the liver, will end up being one of those two nitrogens used to make urea in the cycle called the urea cycle. So I want to emphasize here that glutamine, when it's bringing to the intestine as well as the kidney the ammonia. It's the major transport form of ammonia. It's not a carrier per se like albumin. It is part of this molecule called glutamine. It's the major transport form of ammonia. And it's bringing ammonia either to the kidney or to the intestine. In the intestine, after it's removed, gets in the portal blood, and then picked up by the liver. And the liver will take that amino group and build it into this thing called urea in the pathway coming up shortly called the urea cycle. We'll come back to this issue in the urea cycle in just a few minutes. But the next item is on the upper left-hand part of that figure in front of you, we got to talk about what's happening in the muscle. This slide is taken from the upper left-hand part of that figure in the textbook. This is what happens in muscle when muscle gets broken down under conditions of starvation. In carbohydrate metabolism, we talk frequently about the issue in starvation, how the body needs carbon in order to make glucose. And where's the carbon coming from? Most of it's coming from glucogenic amino acids. And where are the glucogenic amino acids coming from? They're coming from muscle protein breakdown for the most part. So that's what this top of this figure, that's what this figure is all about. So in the muscle, there are a family of transaminases. Now, if there are 20 amino acids, that means there are going to be at least 20 different amino transaminases that will deal with that amino group of the amino acids. Now, all these transaminases require pyridoxal phosphate to remove the amino group from those 20 amino acids. So as I discuss that nitrogen, watch the flow of the nitrogen, as I'll talk about it, as illustrated by a star. So the star is representing the nitrogen of all 20 amino acids when proteins in the muscle are being broken down to get that carbon to make glucose ultimately in the liver. So the issue now becomes when this family of transaminases removes that amino group from all 20 amino acids, the product of that family of transaminases are all alpha keto acids. But then what happens to that amino group? The amino group is put onto alpha ketoglutarate to generate the product called glutamate, the naturally occurring amino acid, glutamate. And then there's an enzyme, sort of a low to medium yield enzyme called alanine transaminase or amino transaminase, same thing. What this enzyme is going to do is use pyridoxal phosphate to remove the amino group from glutamate, and it puts it onto pyruvate this time to make alanine as a product. And then alanine gets dumped into the blood. Now, when alanine gets dumped into the blood, it will not change the pH. And the reason why it doesn't change the pH is because the R group of alanine is a methyl group. It's a neutral amino acid. It will not change the pH. But what's important about the alanine is when muscle proteins being broken down and all that nitrogen is being funneled down to the alanine, what's happening there is alanine is bringing to the liver a source of nitrogen for the body to dispose of it and a source of carbon for the liver to take that carbon in order to make glucose in the pathway of gluconeogenesis. So that's what's coming up next. On the next slide is what happens with that alanine. Here's that alanine coming from the muscle, when muscle proteins being broken down. So the alanine's bringing to the liver that source of nitrogen and the source of carbon to make glucose and nitrogen to dispose of it. So here's the alanine. It gets into the liver with that nitrogen. Now in the liver, that same enzyme that was in the muscle, the alanine amino transaminase, that same enzyme is also located in the liver. And the liver just carries out the reverse reaction. It pops the amino group off the alanine, regenerating pyruvate, which goes into gluconeogenesis. And then the amino group is put back onto alpha-ketoglutarate to regenerate the glutamate. Now, before we proceed here, it brings up a very important issue, and that is, notice what's happening, what your body is doing. It's taking the amino group off glutamate, putting it onto pyruvate to form alanine. Alanine gets to the liver, and the reverse reaction takes place. Why didn't the body just dump glutamate into the blood and get to the liver? Why did it go through this alanine business? And the answer to that question is very simple. It would change the pH. If glutamate was dumped into the blood directly and picked up by the liver, the pH would drop too severely, and you could die because of that. So your body's smart. It knows chemistry. It knows glutamate is an acid. So what it does, it takes the amino group, puts it onto pyruvate to form alanine. Alanine gets dumped in the blood and does not change the pH. Beautiful how that works. But nevertheless, we regenerate the glutamate in the liver. Now, that nitrogen in the liver can come directly from amino acid catabolism by other transaminases and puts the amino group on alpha-ketoglutarate to form glutamate. But most of the time, in the starvation state, it's coming from the muscle, that glutamate. Now, what happens to that glutamate in the liver? What happens is there are two enzymes that want that glutamate nitrogen. Two enzymes want it. Now, these two enzyme names are low yield. Don't go worry about them for the boards. But one of those enzymes can remove the amino group from glutamate, and the enzyme is called glutamate dehydrogenase, shown right here. But again, in my opinion, low yield enzyme. It can remove that amino group from glutamate using NAD, again, low yield, to form alpha-ketoglutarate. But now that amino group will end up right here, end up being one of those two nitrogens to form urea in the urea cycle. The other enzyme is an aspartate transaminase, low yield. But what that enzyme is going to do is remove the amino group from glutamate, puts it onto oxaloacetate to make the 4-carbon aspartic acid. How the amino group could go on to OAA to form aspartic acid. And that nitrogen of aspartic acid will be the other nitrogen used to make urea. So to summarize this issue, because in case the National Board asks, they might ask, where are those two nitrogens coming from, originate, to make urea in the urea cycle? And you answer by saying, one nitrogen comes from aspartic acid, the other nitrogen comes from ammonia. And ammonia itself comes from two sources. Ammonia can come from glutamate, as shown vertically here, or the ammonia can come from portal blood ammonia coming from glutamine. So one more time. That if they ask where are those two nitrogens coming from to make urea, you answer by saying aspartic acid or aspartate and ammonia. Ammonia itself comes from two sources. One is glutamate and the other source is glutamine. Don't worry about the enzymes. If you know where they're coming from, you can deduce just by thought process what enzymes might be involved in case they ask the enzyme. Just know where are the nitrogens coming from.